All right. So good morning and welcome everyone. For today's session, we have two speakers, Dr. Rajiv Jain and Dr. Corey Arnold. Rajiv completed a Bachelor of Medical Science, Master of Science, and a Doctor of Philosophy at the University of Western Ontario. During his graduate studies, he studied the role of peripheral B cell responses in MS, and now studies the role of B cells in CNS of MS patients with Dr. Young. Dr. Corey Arnold attained his PhD in biochemistry and molecular biology at the University of Calgary in 2016. He has since held a postdoctoral fellowship funded by the MS Society in the lab of Dr. Robin Yates. He's studying phagosomal biochemistries and how they direct antigen processing and presentation. So Rajiv will be starting first, so over to you, Rajiv.
Okay, well, thank you for the introduction, Neetu. So today, myself and my colleague Corey here have the pleasure of giving this lecture about laboratory techniques and models used in MS research, and basically how we can use these models and techniques to begin to learn about the underlying pathology of the disease. So the outline for this lecture is, I'll start by talking about the models we use in MS research, which consist of either using human samples or we use animal models to ask questions, which can either be animal models using central nervous system autoimmunity, or we can use demyelination models to ask other questions. After I talk about the models, Corey will then come in and talk about laboratory techniques that you can use in these models to begin asking your questions about the disease. So I'll start by talking again about human samples, but before we get into that, we should talk about some considerations that if you continue to stay in the field of MS research and start to work with human samples, you'll probably have to consider things like this. The first is biomedical ethics, which obviously if you're working with another person, you don't actually want to do anything that would harm them. And so there's a lot of intricacies to ethics, so you'd be worth contacting something like an ethics board if you were to consider something like that. And also consider that if you were to do a human study, depending on what you're trying to do, it can take a considerable amount of time to actually get the ethics approved for the experiments you want to conduct, depending on what you're going to do. You should also consider factors like recruitment. So for example, if you have very specific exclusion criteria, say you were trying to study something like progressive MS, which is relatively more rare, then you might have trouble acquiring patients for your study unless you have a very large clinic supplying you. And also consider the sample that you're trying to obtain, because some samples, like let's say taking peripheral blood, are relatively easy to get, whereas other samples might be more invasive. And also consider that if you're acquiring these samples, you have very little control when the samples will be made available to you, because it's basically just going to be dependent on whenever a patient walks into the clinic and agrees to hand over a sample. And so you could pretty much be called upon any time of the day to have to process the samples. And so it can be, I guess, very laborious having to wait for samples to come in. Lastly, you should consider variability with human samples, because unlike mice, which are genetically all similar, in the human population, there's huge amounts of diversity with respect to genetics. People coming in are going to be at different ages, and also each individual is going to have different lifestyle factors that all would contribute to the variability within your samples. And thus, you're probably going to need larger N numbers with human samples than you would otherwise need with mouse samples. You should also consider the measurements you're going to be making on these samples can also be affected by a lot of factors. For example, circadian rhythms, which is basically how our biology changes over the course of the day, can dramatically affect things. And so if you were to collect a sample from someone in the early morning versus later in the evening, you would find that circadian rhythm by itself is enough to introduce a lot of variability. And thus, if you're working with human samples, you should consider this, because if you're collecting samples at different times of the day, you're already introducing a lot of variability. Also consider the amount of time it takes for you to process or store, or the amount of time your samples will be stored, because this can affect things like the viability of your samples, or the degree to which biological molecules will be degraded. And lastly, if you're assaying your samples on a machine on different days, it's possible that the machine will have variability over time, and that should also be taken into account. Okay, so as far as human samples go, the one type of sample that you're most likely to get your hands on would be a tissue section. And reason for this is that tissue sections are usually donated by individuals post-mortem. And so these do not take a terrible, or they're not really invasive. It's pretty easy to acquire these, and so most labs can get their hands on these. The way this works is basically you'd be sent a block of tissue, and this block of tissue would then be cut into smaller sections that you can then use in subsequent analysis. That analysis can be a variety of different things. I'm not going to be talking about the specific methods that you can use, because that's what Corey will be talking about. But in general, you can use these for things like RNA analysis to look at the expression of genes in your sample, or you can use it for histology to look at the pathology going on in a tissue, or look at the cellular composition in a MS lesion, for example. So an example of this is this section from Dr. Deepak Kashuk, where he basically did a Luxol fast blue stain and an H&E stain on this MS section. And what you can see is that whereas you have even blue myelin staining all throughout the white matter of this MS section, you don't see it right here, where this would basically represent your MS lesion. And so using these stains can be really valuable to basically identify where pathology is happening in MS, but also to learn about the cellular composition, as I alluded to, which Corey will talk about later. Another sample that you can get your hands on would be the peripheral blood of a person. And so peripheral blood isn't too invasive, so there are some complications that can occur, but for the most part, it's not too hard to get a study approved where you'd just be taking peripheral blood. And the way this would work is once you have the tube of blood, you would then centrifuge it to separate it into different layers. Typically, this bottom layer will consist of your red blood cells, and for the most part, you just throw those away because you're not really interested in studying the red blood cells. Instead, usually what you're going for are these top two layers, which would consist of your buffy coat, which would contain a lot of your immune cells or peripheral blood mononuclear cells. And with that, you can do a lot of different assays with. You can, for example, do flow cytometry or single cell sequencing techniques, or you could use them in ex vivo in vitro cultures. And this gives you a lot of flexibility with what you can actually test with these immune cells that you're isolating with the blood. But the other component you would also get is the plasma or serum from these individuals. And this is basically the fraction of blood that contains all the proteins within it. And so this would consist of things like antibodies, cytokines, or other soluble proteins, which can be useful for identifying things like biomarkers for MS or looking at the specificity of things like antibodies. And thus, peripheral blood is really valuable for the study of MS. The other sample you could potentially get your hands on would be cerebral spinal fluid. And so cerebral spinal fluid is basically the fluid that permeates our central nervous system and thus contains lots of things like myelin proteins and a lot of the immune cells that you would find in MS. In fact, immune cells are increased in the CSF during MS as well. So CSF is somewhat invasive to actually obtain because you have to do a spinal tap, and this isn't a particularly pleasant procedure to undergo. It can be quite painful, and so this would definitely be a harder sample to get your hands on. But it's very valuable because, like I said, this is literally allowing you to get a snapshot of what's going on in the CNS of the individual that you're sampling. In particular, what you would do with CSF is you would again spin it down like the blood to separate your immune cells from the rest of the fluid portion, which would be the CSF. The immune cells, you generally are limited to single cell analysis because you're really not going to get a lot of immune cells from one of these spinal taps. You might get at best something like 3,000 cells. And so you're highly limited in the sort of analysis you can actually do with them. But with the CSF, you can again look at things like antibodies, cytokines, and other soluble proteins to look for things like biomarkers, which could be really valuable. So the pros and cons of using human samples. So some of the pros involve that you're studying the actual disease and not a model of it. And so this is the closest you can get to really getting true information about what's actually going on in MS, and can be a very good predictor of clinical aspects of the disease. Human samples are also needed to validate phenomena that you might see in animal models. And so even if you have really good results in your animal model where you have something really cool, unless you can validate it back in the human setting, then that's really going to take back from the impact of your work, because if it's only an artifact of the animal model, it's hard to say that it would be important for the study of the disease. There's a lot of cons though. So first of all, if you're working with tissue samples, as I alluded to before, these are typically given post-mortem or after the individual has passed away. And as a result, these samples are highly biased towards the end phase of the disease. And so it's very hard to learn about the early phases of MS from these tissue samples. Furthermore, it's also usually not easy to study the tissue of interest if you're working with live individuals with MS, because obviously you can't take out the brain of someone who's currently still living. You're kind of forced to work with what's available, which isn't always representative of what's going on in the tissue. Furthermore, if you're going to use cells from MS patients, you're pretty much going to have to use them in some sort of in vitro protocol, which would basically introduce an artificial element of the basically taking them out of the in vivo environment and putting them in a situation they shouldn't otherwise be in. And also, lastly, there's a limited number of techniques that you can actually do with human samples, and thus it can be very difficult to establish causation using just purely the human samples. And instead, establishing causation is much easier in the animal models. And so the first of which of these animal models that I'm going to talk about are the models of central nervous system autoimmunity. So why do we care about autoimmunity? Well, as we've talked about before in this lecture series, there's a lot of evidence that suggests autoimmunity is a core component of MS. And this is supported by the fact that MS therapies target the immune system currently, that genetics strongly implicates the immune system, and that immune cells are found within MS lesions, all of which suggests that autoimmunity is a prominent, or has a prominent role in the disease. Fortunately, modeling a CNS autoimmune attack isn't terribly difficult, because all you really need to do is trick the immune system into attacking the CNS. And this can be achieved by combining a strong adjuvant, which is basically an immune stimulant, with a protein derived from the central nervous system, and then immunizing the animal with this concoction. And what that will do is it will basically drive a peripheral immune response, predominantly, or most importantly, it will drive a CD4 immune response. And these CD4 T cells can then move into the central nervous system and basically contribute or start lesions and ultimately initiate the disease in the animal. So the model we typically are using to study this, or the autoimmune component of MS, is experimental autoimmune encephalomyelitis, or EAE, to which this EAE model can be broken down into a lot of different forms. One of the major forms would be the active version of the disease. And the way you induce active EAE is you combine complete fruit adjuvant, which would represent our strong adjuvant, along with myelin protein, as I alluded to before. The protein that you combined with this adjuvant can be either just whole CNS homogenate, so that'd be basically grinding up the central nervous system of an animal, or full-length myelin proteins. So this would be focusing on something specific like the MOG protein or PLP, for example. Or you can take just the portion of these myelin proteins that is capable of inducing a CD4 immune response and immunize with that. And so examples of common peptides used to induce EAE are the MOG35-55 peptide, the PLP peptides, and MEP peptides, where all of these represent this minimum amount of myelin, each of these represents the immunodominant epitope in each of these antigens for stimulating CD4 positive T cells, which is basically the minimum requirement for inducing EAE. When you induce EAE, you also combine it with pertussis toxin generally. And so the way this works is on the day of immunization, where you're giving the myelin antigen with your adjuvant, you're also going to give a pertussis toxin injection, usually about 200 to 400 nanograms, on the day of an immunization, but also 2 days later. This will ultimately lead to the induction of EAE, which is typically is scored on a lot of different scales, but the most common of which is this 5-point scale. And in this 5-point scale, it's basically describing ascending paralysis, where at 0, the mice are healthy. At a score of 1, that represents tail paralysis. 2, that represents hind limb weakness. 3 would represent hind limb paralysis. 4 would be forelimb paralysis, and 5 is pretty much where you shouldn't let your mice ever go to, but that would represent the point just before death. So in a typical disease curve, what you'll find is there's this pre-symptomatic period that's followed by the first day that one of your mice ends up developing any signs of the disease, and this is disease onset. And so for this particular experiment, it would be at day 7. If we then let the disease continue, we then end up with a point where the disease is at its worst on average across all your mice, and this would be peak disease. You can then let this go on even further, at which point you end up at chronic time points, at which point the disease curve will do one of two things. It will either come up, hit this peak, and then fall down completely to a recovery phase, or the mice will stay chronically sick, depending on the model you use. So some fun facts about the history of EAE. So EAE was actually first done in humans by accident, and this was as a result of trying to make a rabies vaccine. And so they basically took the spinal cords of those infected with rabies and had combined it with an adjuvant to try and basically make people immune to rabies. And while this probably was successful, the problem was the people immunized with the spinal cord homogenate also ended up developing EAE. So obviously not a very effective immunization. And also, generally speaking, there was a progression from relatively crude CNS homogenates being used in experimental protocols, whereas now the most common protocol involves immunizing just with the CD4 stimulating component, and so a relatively more simplistic model. But nowadays, it's probably more common to use the right model for the right question. And so there are a lot of variations on active EAE that allow you to ask questions specifically about what you're actually, or certain models of EAE are better for studying certain things. So for example, different mouse strains will develop different forms of EAE. So the C57 Black 6 strain typically develops this sort of curve shown here, where there's an initial phase of demyelination and then they recover. Or they have an initial phase and they stay sick. The SJLJ strain, however, is a little bit different. They usually develop this relapse-remitting type of curve, which is advantageous if you're trying to study RRMS sorts of pathology. You should also you should bear in mind that Different myelin proteins can induce slightly different forms of EAE. And also there's a difference between immunizing with whole protein versus just the peptide fragment for inducing CD4 immune responses. So an example of this I'm showing on the right, where basically if you immunize C57 Black 6 mice that are either deficient in all B cells or just have a wild type repertoire, what you find is immunizing with just the peptide results in worse EAE if B cells are not present, suggesting in this model that B cells have a regulatory component. But if you use another model, such as immunizing with whole MOG protein, you see a very different sort of phenomenon. In particular, you find that immunizing with whole MOG protein, you absolutely require B cells to be there. Otherwise, the mice do not develop disease. And so you have, depending on the model you use, it can be either better or worse for studying certain subsets of cells. So for example, immunizing with just the peptides I alluded to earlier will induce CD4 immune responses and also myeloid immune responses, but not appreciable B cell or CD8 immune responses. Whole protein immunization can induce all of these cells, but doesn't appreciably induce a CD8 immune response. Now, the one problem with whole protein is it's a lot more expensive and cumbersome to work with, and so there's definitely a trade-off. So depending on what you're trying to ask, it may be advantageous just to use the peptide. The other type of our form of EAE that I'm going to talk about is passive EAE. And so what passive EAE is, it involves basically transferring pre-activated autoreactive T cells into an otherwise naive animal to induce EAE. And so an example of this is shown at the bottom, where basically you would immunize a starter mouse with basically one of your myelin proteins, whichever is your favorite, and you'd try to induce EAE in this mouse. But then before disease onset occurs, you basically isolate the T cells from this mouse and plate them in a petri dish and re-stimulate them with the same antigen you primed with. You can then also add cytokines if you want. We'll talk about that in a second. But after 4 days of culture in vitro, you can then transfer these in vitro cultured T cells into naive animals, so no other manipulation other than transferring the T cells, and this mouse will develop EAE. So why would we care about using passive EAE? Well, first of all, it can be induced with CD4 cells or CD8 cells. And so if you want to study CD8 biology, this is one model you could potentially do it with. It also subverts the need to immunize this animal that you're inducing EAE within, because immunizing by itself changes the biology of the mouse considerably. And so to get a less biased or a less biased immune response, it may be advantageous to skip this immunization component. You can also easily manipulate the genetics of this system. So you can manipulate the genetics of the T cell or the recipient mouse, giving you a lot of flexibility with the experimental, how you set up your experiment. And also you can control the polarization of the T cell you put into this mouse. And the reason this is important is because if you were to say add cytokines to polarize towards a Th1 cell, this induces a disease that's more polarized towards the spinal cord. Whereas if you add cytokines to skew towards Th17 cells, this generally leads to more brain inflammation. And so if you want to study brain inflammation versus spinal cord inflammation, this could be an important variable that you can manipulate. There are also other forms of EAE that I'm not going to talk about in detail, but I thought I'd throw in a slide so you can at least see them. So there are viral forms of EAE wherein basically a virus would infect the CNS and basically the immune system would then clear up the virus, which is great because it induces a very potent and diverse immune response, but is artificial in the sense that we don't believe MS is driven by a virus infection of the brain. You can also use non-human primates, which is a great transition from your benchside research into moving a potential treatment into the clinic. And you can also use spontaneous models, and this would again circumvent the need for immunization, which could again potentially bias immune responses. So the pros and cons of EAE. The pros would consist of the fact that this is a established preclinical model for MS therapies and is widely used, and the pathology is well known, and thus is a great model for studying immune-mediated inflammation of the central nervous system. There are also a lot of variations on EAE, and thus if you have a specific question you want to ask about a specific cell type, there's probably an EAE model that you can use to ask that question. The cons of this is that EAE can vary substantially between institutions and also potentially over time in a lab, and thus keeping things the same over time can be very difficult. EAE induction can be highly variable between mice. And so if we were to go back to these disease curves I alluded to, these look very nice, and it looks as though the mice would be developing a similar course of disease, but the reality is there would be a lot of heterogeneity. Some mice would develop disease faster, some later, some would develop less severe disease, some more severe, and also some mice would, I guess, go into recovery phases, whereas others might have a secondary attack or chronic attack. And as a result of the variability in EAE, it can make studying precise timings in the pathology very difficult. EAE also doesn't really reproduce the neurodegenerative aspects of MS, and thus you can't really study this process. And it's also hard to generate a progressive model of EAE because generally speaking, the mice will either get really sick and then just stay at that same level of sickness, or they will get sick and just recover. Which brings us into the advantages of using demyelination models. So why would we care about using these demyelination models? Well, currently MS therapies pretty much just target the immune system. We've kind of neglected the neurodegenerative and repair aspects of MS, where repair in MS would pretty much come in the form of remyelination. And so remyelination is a process wherein basically oligodendrocyte precursor cells are recruited to the site of damage or where there's demyelination. These oligodendrocyte precursors can then differentiate into mature oligodendrocytes and remyelinate the myelin. This is a really important process if we want to help repair the damage that is occurring in MS patients, but is difficult to study in EAE predominantly because of this, because the animals do not develop a uniform disease. And this is where the demyelination models really shine. So I'll be focusing on two demyelination models, the first of which is cuprizone. So cuprizone, the way this model works is it basically uses a copper chelator that you basically put in the food for your animal of choice. And this copper chelator will basically drive oligodendrocyte death in the central nervous system through a process that we don't really understand appreciably. The demyelination in this model is typically studied in something like the corpus callosum, although it can be found in many other sites of the brain. And in this model, the mice are again fed cuprizone in their diet, and this leads to a progressive demyelination in these mice until the diet is stopped, allowing for remyelination. And this is one of the big pros of this model, is that you can control when you want the remyelination to happen. This model is really useful for predicting, or because it has a very predictable course of demyelination in pathology, allowing you to basically use very precise timings in this model. The length of the demyelination period can be modified pretty easily as well. And so at the bottom, I've shown you an example of the pathology where basically in a healthy mouse in the corpus callosum, you would have very nice myelin staining as seen by this POP. Whereas when you induce demyelination, you can see quite clearly that you lose demyelination in the corpus callosum. But if you allow these mice to recover, you then get remyelination back again. So typical, or the way the cuprizone model is used, generally speaking, is again, you're feeding them cuprizone for about 5 weeks or so, depending on what you're trying to study, where again, we know how this demyelination works in a good amount of details, and we know when the oligodendrocytes are dying, we know when CNS resident cells are becoming activated, and when oligodendrocyte precursor cells are trying to repair this damage. And so because it's well defined, this makes it a really nice model. We also, again, can modify the time of demyelination, where if you use relatively more acute demyelination periods, you have a robust remyelination. Whereas if you're using something like more chronic demyelination, you get less robust remyelination. Could be a nice model of when in MS you have chronic demyelination and thus have a much harder time of actually remyelinating in this scenario. And as a fun fact, cuprizone can also be converted into EAE. So the STICE lab here at U of C has shown quite nicely that if you do a brief cuprizone induction period followed by an immune boost, where the immune boost is the CFA component that we use in the EAE induction, you can actually convert this cuprizone model into a model of central nervous system autoimmunity, wherein basically damage to the brain leads to a secondary immune response that then invades the central nervous system. The other model I'm going to talk about is the toxin injection model, which basically involves literally doing surgery on a mouse and injecting some sort of white matter tract with some sort of toxin that induces demyelination. And so this model can be done pretty much anywhere in the central nervous system that's accessible by surgery, although it's typically done in the spinal cord for ease of access and the corpus callosum because it has a well-defined white matter tract. A common example used in this model is the lysophosphatidylcholine, or LPC molecule, which when you inject it into the spinal cord, you get a very nice demyelinated lesion. And then if you wait over time, you can get remyelination, where basically LPC works as detergent because it's a lipid and basically destroys the myelin at the site of injection, but then the axons are left somewhat intact and thus can allow for remyelination. This model is again useful because it has this predictable course of demyelination and pathology. Potential bonus is also that it doesn't have an adaptive immune response, and thus if you're trying to focus purely on just what's going on with the biology of remyelination, this can be a really nice model for specifically looking at those processes. And also that you can pretty much inject whatever you want into the spinal cord, so you're not limited to this LPC model, and thus it's very flexible in that regard. So the potential uses or pros and cons of this model. This model allows for consistent quantification, or these models generally allow for consistent quantification of demyelination or remyelination. And this is really useful when you're trying to assess things that will actually influence remyelination. It also has a predictable course, which allows precise study of how the CNS reacts to damage and how the mechanisms of repair actually work, and that these models are very flexible. Cons are predominantly that it does exclude the adaptive immune system, and thus it makes it somewhat hard to extrapolate these results to MS. And also that these models are using somewhat artificial methods of damaging the CNS that are unlikely to be a part of the MS pathology, although this does depend on what you're using to induce damage. Okay, so in summary, I've told you a lot about the samples that you could potentially use to conduct MS research, and this is basically going to be your toolbox that you're going to use to basically answer questions about MS. And now I'll hand this over to Corey, who will talk about the methods you can use to actually use in these models.
All right, thanks, Rajiv. I'm trusting everybody can hear me. So yeah, I'm going to talk about some of the more technical aspects of MS research and what we do in a basic lab environment. A couple quick disclaimers. One, you might see a bunch of squares on my text, and that's because through some technical issues, I had to create my talk on the online version of PowerPoint, and this is just kind of how it translates in the presentation mode, so I apologize for that. Two, there are hundreds of thousands of techniques out there and variations upon a whole bunch of them. I'm going to touch on the ones that I think are probably most appropriate to what summer students would use or would be using should they be in the lab this summer, barring any COVID nonsense. And so if your favorite technique doesn't come up, I'm sorry, but I had to pick and choose some of what I consider, I guess, my favorites and some that are more applicable to MS research. So,
Sorry, we can't see your slides.
Sorry?
We can't see your slides.
Oh. One moment.
Rajiv, please stop sharing so that...
He did. We are good. My mistake. Okay, there's the squares. Great. Let's kick this off. Okay, so in setting this up, I decided to sort of do this from the smaller stuff to the bigger stuff. So assays that we use to look at molecules like DNA and proteins, and then assays we use to look at cells on a whole or cell populations, and then finally things we use to look at gross tissue morphology and so forth. So we're going to go kind of from small to big as I move through this. So one of the most common things that people use to study in the lab, one of the most versatile assays or techniques is called PCR, and most of you, I'm sure, know exactly what this is, but its main purpose is to exponentially replicate targeted DNA segments, and it does this by co-opting a DNA polymerase from a thermophile bacteria, and this polymerase can recognize double-stranded DNA created by short primers to target your DNA sequence you want amplified, and then extending that into double-stranded sequences. And by cycling the steps of denaturing, annealing, and extending, you can create vast amounts of DNA of a particular sequence that you specify. And you can sort these based on size by running them through an agarose gel. Now, like I said, PCR has a lot of different potential applications, cloning, sequencing, genotyping, and so forth. But the one I want to focus on is called quantitative PCR. And quantitative PCR essentially lets you relatively estimate or figure out how much starting template you had. This is very useful for looking at expression of genes, so how much mRNA is created from a certain gene at the start compared to another gene, and whether it's different between cell samples and so forth. And so to do this, basically they use a dye, either a dye or a probe during the PCR cycling steps, and that dye will fluoresce when the DNA is double-stranded at the end of the extension portion. And the more DNA you have that is double-stranded at the end of that portion, the more fluorescence you're going to have, and so you're going to see an increase in fluorescence signal. Or you could use a probe, and you can use a probe that's specific to the DNA segment that you want to look at. And that probe is normally quenched until the polymerase comes through and frees it up. And so the more template that the probe is freed up from, the more fluorescence you're going to have. The dye tends to be a little simpler and usually a little more easy to, or a little cheaper, so that tends to be one that people use, but the probe-based method gives a lot more specific signal. And so when you're doing your assay, you're going to see a chart that kind of looks like these. Let me see if I can bring up my pen here. Oops. I'm not sure if people can see this, but on the left here, you have a curve that indicates basically what's going on during your PCR. And you set an arbitrary threshold, and the cycle at which the sample crosses that threshold is known as your CT. And once you figure out all your CTs for these samples, you can determine how much, you can calculate back how much starting sample there was and compare them to one another. Now, this is assuming that the PCR reaction is 100% efficient, but there is, I've just brought up the title of a paper here, a reference to a paper here that kind of looks at some other algorithms to include, assuming that your PCR sample isn't 100% effective. However, for most people's purposes, using the delta CT method covers what they're trying to show. Another way to look at RNA expression, while qPCR is useful, you only look at a couple of genes at a time. Sometimes you want to look at the global fingerprint or analysis of RNA transcription. So what transcripts are going on within a whole cell at this particular time. And so this is done by creating a library of RNA segments converted back into DNA, and you add these adapters, which are denoted by the red and blue on the end of those little short segments at the right there. And then you can sequence these, and you get hundreds of thousands of sequences or reads that come out of this, a huge data set, and you align these reads back to a reference RNA, full sequence of RNA or complement of RNA that is known. And the number of reads that come back will tell you how many of each RNA segment there was to begin with. And you can plot that in a heat map, which is shown on the left, and will tell you relatively within each condition, which is shown at the bottom, whether an RNA is more or less represented in a particular sample. And these RNAs can cover a whole bunch of different, you can look at any number of sequences because it looks at your whole transcriptome. And a very popular and more common thing, or something that's being done more and more common these days, is looking at single cell RNA sequencing. So rather than looking at a bunch of cells within a tissue as a sample, this can be done on individual cells, and each cell can give you a fingerprint of what it's expressing. And then these cells can be clustered based on how similar their RNA fingerprint is, and in this type of plot here, which is called the TSNE or a TSNE. And people can assign what kind of cells they are based on their expression profiles, or redistribute how they want to group them based on their expression profiles. So that's RNA, both at kind of an individual and a global level. But as most people know, or you may not know, how your RNA is being expressed does not necessarily correspond to how your protein is being expressed. So it's also good to look at protein expression within samples as well. And by far, the gold standard for looking at this is the Western blot. And this can detect proteins that are separated based on size, very similar to what we saw with the DNA agarose gel on the first slide. However, you have to also transfer proteins separated based on size from a gel onto a membrane, usually a PVDF or a nitrocellulose membrane. And when that protein is present on the membrane there, you can see in the bottom left corner of this left diagram, the protein is available to be immunolabeled by an antibody that recognizes it specifically. And then that antibody, through a chain of primary and secondary antibody labeling, an enzyme conjugated in the mix will transfer a substrate to a signal that can be detected on the membrane, and you get these bands showing up that are showing up at the whatever the size of protein you're looking at is. This immunolabeling technique is used very extensively in a bunch of different things, and will be coming up again and again as I go through the talk. And we can just see on the right side here an example of Western blot in action. So this is a MOG protein, and Western blot can sometimes be used for quantification, but it's only ever semi-quantitative at best, but it's still a very common practice to use. So the intensity of bands, if you look at the anti-MOG in the middle lane there, you get differences in expression or the intensities of these bands, and these can be interpreted as a graph known as densitometry. And another way to look at proteins on a blot, instead of using a chemical reaction to create a signal, what is gaining a little bit more traction is the use of fluorescent antibodies and using a fluorescent Western blot. And one advantage of this is that you can actually look at two different proteins at the same time. Whereas if you're only using typical chemical immunolabeling, you can only look at one protein per blot. So Western blots are great, and many people use them. Oh. But ELISAs are also great, and they are very good for looking at proteins in a more quantitative fashion. So this is generally done in a 96 well or a 384 well plate, where you have the antigen, the protein that you want to detect, bound to the bottom of the plate, and then you introduce antibodies recognizing this in however fashion, either as a direct ELISA, where you just have a primary antibody and the substrate conjugated, or the enzyme conjugated to it, or you do something similar to what we saw with the Western blot, where a secondary antibody is used. This is more useful for being flexible in terms of what you can use your primary antibodies for and your secondary antibodies. And instead of having to conjugate an enzyme to every single primary antibody you use, you can just reuse a secondary antibody that recognizes all these different primary antibodies. There are sandwich ELISAs, which instead of binding the antigen to the plate, you use a capture antibody, and this is a very highly specific way of looking at things. Or you can use a competitive ELISA, which is very good for looking at small quantities of protein within your sample. So those are two ways to look at, again, one or two or maybe a small subset of proteins at a time. But if you want to look at the global protein identification within a sample, mass spectrometry tends to be the way to go. Mass spectrometry is not only used for proteins, but it is very useful for proteins. And basically it takes your cell sample or whatever sample you have, it will ionize them, and it will shoot them through a tube and apply a magnetic field. And these ions will bend based on how charged they are and how heavy they are. And this will be detected at the end of that flight path. And where it hits on that detector will come up as a peak on a mass spectrum, which you can see in the middle there. And so the more ions you have hitting in a particular region of the detector, the higher the peak will be. And so the height of that peak roughly translates to how abundant that ion is in the first place. And this can be translated back to which proteins, once you know the size and charge of these things, it can be translated back to which proteins are present. Now, most people these days use a further fractionation method, which is called tandem mass spec, or MS2, and it provides a more fleshed out spectra. And so after the first peak is generated or identified, you do a further collision or fractionation of that particular sample or peak, and that will create a subset of peaks, which gives you more information about what's making up that particular ion. Now, that's all well and good for identifying proteins, but if you want to actually quantify things and differences between samples, you have to introduce labels into these things. And so to do that, there are a bunch of different methods, but two that I like and are very commonly used is one is called SILAC, which is stable isotope labeling of amino acids in culture. And basically you introduce either heavy or light arginines or lysines. The cells will then incorporate those amino acids into the proteins they make. And after they've grown up a bunch, you can collect that protein and mix the samples together and run them through the mass spec. And this will create a slight shift on the mass spectrum that can be detectable and tell you just how much of protein came from condition A versus condition B. And another way is to do isobaric labeling. And so this is where you label your proteins after you've collected them. And these isobaric labels, or these tags, are initially the same weight. So they'll come through the first spectrum as the same weight. But when that second fractionation happens, the tags split and they actually are differentially weighted. And so will come up on your second MS spectra as differently labeled. And you can determine which samples came from which. So that's kind of a lot of things that we use to look at molecules, DNA, proteins, RNA. But we want to get there are also times when you're going to want to know information about a full cell population. What are they expressing? What kind of cells are there, and so forth. And so you can use flow cytometry, and this is a favorite of immunologists, where you immunolabel cells. So the same thing that was used to detect Western blot. You use antibodies against various, usually cell surface proteins, and these are conjugated to fluorophore. You pass it through a laser excitation, and based on how much light they deflect, how much light they let through, and the particular excitation of their antibody, you're going to get what's this dot plot that you can see on the side here. And you can subdivide the populations that you find in these dot plots through a method called gating. So you can essentially select a very particular portion of that you want to further look at and change which signals you're using to look at each population. And so in the example on the right here, people have taken the lymphocyte fraction from that very first top left graph. They're looking at cells that are expressing CD4, so these would be T helper cells. And then within that set, they've further subdivided what they want to look at and are eventually able to suss out of those T helper cells, which ones are Th1 and Th2, and which ones are Th17, and to some degree their activation state. So not only is it going to tell you what kind of cells you have, but it can also tell you how those cells are activated and what their biological state is. So flow cytometry is a huge mainstay, but it does have some limitations, and a lot of those are overcome by something called mass cytometry, otherwise known as CyTOF. So mass cytometry is kind of an amalgamation of flow cytometry and mass spectrometry. And you immunolabel cells instead of with fluorescently tagged antibodies, metal tagged antibodies, and then subject these cells to a mass spectrometry workflow. And so that has a lot of cool words when you're talking about mass cytometry. You nebulize cells, you aerosolize cells, and that gets them into a single cell proportion. So you can actually do this one cell at a time, very similar to RNA-seq, single cell RNA-seq. And these things are passed through instruments that remove low mass ions. So a lot of the organic ions are removed, and you're only left with the metal labeling ions. And these are subjected to mass spectrometry and create a fingerprint of what those cells were expressing on their outside. And as you can see, the readout is very similar to what we saw with the RNA-seq. You get a TSNE graph on the bottom right there that will tell you which cells are similar, which cells are different, based on what they've expressed on the exterior of themselves. So just sort of talk about why mass cytometry is a little bit, what the advantages are. During flow cytometry, the fluorophores have a lot of overlap just based on how the physics and the optics of the light work. And so you get a lot of false signals, and there's a very limited range of which you can label cells with. However, when you're doing mass cytometry, it's a very discrete spectra that you get from these metal ions that you can label with, and there's a lot more of them. So you can get a lot more information out of this. However, it tends to be more expensive and technically complex. You got to balance yourself. So taking a step up, we've kind of figured out what our cells look like. What do they look like altogether? What's going on in a tissue sample? What's going on in our actual samples at a very gross or morphological level? And so a very easy and straightforward way to do this is to cut it up and stain it or immunolabel it and look at it. So when you're sectioning tissue, there's a couple ways to do it, and two of the more common ways are to use paraffin sectioning, where you embed it in a paraffin wax. It's very stable and easy to section, but you need to fix your tissue, and there's a lot of extensive processing that goes along with it. So if you want to use fresh tissue, you can cryosection it. This gets embedded in basically a frozen gel, and you can section it so that the tissue is still fresh. However, you have to be very careful. It's difficult to do, and your hands are always in this very cold sectioning machine. So that's a negative. So once you've sectioned this tissue, there is a wide range of things you could do to look at it. Rajiv mentioned these early on in his portion of the talk, and these are two of the most common histological stains used for MS or EAE studies. The first being hematoxylin and eosin. This is used almost across the board in terms of histological assays. The hematoxylin will identify nuclei within a tissue sample, and eosin will look more at the cell cytoplasms and whatnot. And you can see just, I'm just going to also talk about the other stain, Luxol fast blue, that he mentioned. And this stains myelin. This is attracted to bases within the myelin sheath, and it comes up blue. And you can see on the left, the 3 ones are control samples, but on the right, you have 3 samples from an EAE affected, I believe it's brain. And you can see at 4 weeks, you have a huge infiltrate of what would be the immune cells. And at that same area, you're seeing a degeneration of the myelin sheath. And this is all just shown by simple, quick chemical stains of tissue. But if you want to get more specific as to what you're looking at, you can use immunohistochemistry. And again, we're going back to how antibodies recognize proteins. And you can use antibodies conjugated with either a colorimetric producing enzyme, and this creates a stain in the area where they are localized, or you can use fluorescent antibodies, and they create very pretty pictures of colorful cells. And a good advantage to using immunofluorescence is that you can overlay 2 different antibodies and see what kind of co-localization is happening. So you can get all these images from tissues and whatnot, or you can create these tissues and the staining, but you have to image them in order to look at them. And so that's where the use of microscopy comes in. Microscopy is all of the methods and instruments we use to visualize images and image cells and tissues. There's 4 branches. I'm only going to talk about the first 2, optical and electron, because these are more commonly used in research. Optical is the use of light or laser to illuminate a sample, and electron microscopy uses electron beams that are deflected by samples and detected. So with optical microscopy, most of what you'll find in a lab is a compound microscope, which essentially means it's a mixture of lenses and mirrors to redirect and focus light. I'm sure most of you have seen a dissecting scope where the light source and the objective lens are on the same side, or inverted scopes where the light source and the objectives are on opposite sides of the sample. And this is very good for looking at cell cultures or slides. And these things are often equipped with cameras to take pictures, either bright field pictures of stained or live samples, or you can look at fluorescent images of samples that have been immunolabeled with fluorescent antibodies. And while basic compound microscopes can take some pictures, we have definitely advanced our abilities and technologies of microscopy to take very nice, very clear pictures. 2 of which I'll talk about are confocal and 2-photon microscopy. The confocal improves your image by using a pinhole in front of the detector to remove any non-focused light, and so the only light you're seeing coming through is that which is on the focal plane that you're looking at. And 2-photon kind of expands on that by using excitation that on its own a single wavelength can't excite a fluorophore. However, 2 of them can, and only where they meet, which is on the focal plane, will they show fluorescence, and kind of that concept is displayed on the right in the middle there. And so that actually increases the ability of the microscope to look through tissue and has a much deeper penetration of tissue. And just on the left here, a couple of examples of images taken with confocal and 2-photon microscopy, and they are very beautiful. And finally, electron microscopy has some very, very cool images associated with it. The simplest way I can describe this is with scanning electron microscopy. Electron beams are shot at a sample and bounced off, and it's detected where they bounce off and can be rebuilt into an image. So here's an image of a macrophage on the left there, done by scanning electron microscopy. And then transmission electron microscopy, where the electrons are shot through a very thin slice of sample, and then where these things are not making it through, it becomes more dense and dark. And that is a TEM electron micrograph of a macrophage as well, and you can see all of its lysosomes and phagosomes there. So that was a very quick run through of a very small number of techniques that are available to you. And I hope you picked up on while I was talking that while you're doing assays and whatnot, you need to make a lot of considerations into which technique to use. Now, it might be very obvious that you can't use microscopy to do PCR reaction. However, what kind of PCR are you going to use? And so whenever you're going to pick a technique to answer a question you might have, you want to make sure you know specifically what question you need answered. the availability of the reagents that you have, as well as the availability of the equipment. How much is this going to cost? Is this going to bankrupt PI? How much time is this going to take? Are you going to spend 3 weeks just doing one thing? How difficult and reliable is it to work? And also, how relevant is this technique? Are you using something that was established in 1984 and hasn't been used since? So all these things to take into consideration. Thanks for your time. And if you guys have any questions, hopefully we have a couple minutes to just quickly answer.
Very well done, Corey and Rajiv. Excellent. Questions from anyone? I see, in the chat box that there are